happy to have the opportunity that I've never talked about in public before. And some of it will have a little bit of preliminary data feel, and so I'd be interested to hear your feedback about some of the studies I'll be discussing. I don't know if we had to do this here, but I got nothing. So as an introduction, I'm sure you're all generally familiar here, but I want to place into context the GnRH neuron, which is really the topic of today's talk. In the hypothalamic pituitary gonadal axis, the GnRH neuron is the final common output from the brain, so it's a final neural output regulating the pituitary gonadotropes, which secrete LH and FSH into the circulation. These in turn stimulate the maturation and development of the gonads, testy in male, ovary in females, and also stimulate the synthesis and secretion of the gonadal steroid hormones, males testosterone, females estrogen and progesterone primarily. These then can exert feedback regulation at both the level of the pituitary and of the hypothalamus, and here I'm pointing toward the GnRH neuron, not at it necessarily, but and in males, this is always negative feedback, and this is just homeostatic regulation of testosterone levels. And in females, we have that more interesting addition of positive feedback, which is required for the elaboration of the preovulatory gonadotropin surge of LH and FSH, primarily LH, that triggers ovulation. So both negative and positive feedback then are essential for proper reproductive function, and the regulation of this feedback is of great interest. About 12 years ago, we added a new member to this axis, the kisspeptin neurons, and they lie sort of upstream of the GnRH neuron and potently regulate GnRH neuron secretion and GnRH expression, and have been shown to play an essential role in puberty and ongoing reproductive function. And there's also convincing evidence that they play an important role in both negative and positive feedback, and I don't think there's a ton of debate on that. They do play a role in that. So the kisspeptin neurons then have estrogen receptors, primarily ER alpha, and can mediate some of both negative feedback in males and females and positive feedback in females. Okay, let's see here. Okay. So I would say that the dogma in the field has always been that GnRH neurons do not have estrogen receptors, and actually also they weren't reported to have testosterone receptors. And these studies were first published, I think, around the late 80s. I know Don Pfaff's lab seemed to have some autoradiography studies, and so all the early immunohistochemistry suggested that the receptors were not there. I would say that these studies have recently been challenged by the identification then of this isoform of the estrogen receptor beta, which provided a new potential receptor, estrogen receptor, in GnRH neurons. And a number of groups have shown that ER beta is expressed in GnRH neurons, and including, I think, Gloria and Isfan are my co-session speakers today. And a number of studies have shown that in GnRH neuronal cell lines, there's beta receptor, but also some have shown that there's alpha receptor. So this could be due to the differences in cell lines versus the neurons in vivo, or it could be that you have improved sensitivity in cell lines because you can get lots of material to make your measurements, and maybe there is low levels of alpha in vivo. And it could be that we're catching, and I'll show you some evidence for this, developmental snapshots in some of these cell lines, and maybe you see alpha at certain times. So clearly estrogen receptor is important for reproduction. In mice, we have inactivating mutations of both ER alpha and ER beta, and they result in various degrees of severity of reproductive dysfunction. In males and females, knockout of ER alpha results in infertility. In males, if you knock out ER beta, there's no report of infertility. However, in the females, it's a little confusing. There are sort of a spectrum of reports about the reproductive function of ER beta knockout mice. These range from disrupted preovulatory LH surge capacity, differences in ovarian steroidogenesis, folliculogenesis, and variable infertility in ER beta knockout mice. So a number of more recent studies have used conditional knockout technology to knock out ER alpha and some ER beta in different compartments of the body and different tissues, including the different levels of the HPG axis, including knockout of ER alpha and ER beta in the brain, ER alpha in the pituitary, ER alpha in different compartments, different cells of the ovary, but little has been done so far looking directly at the level of the GnRH neuron and the effects of ER alpha and beta. And this is probably a result of the idea that there aren't really these receptors in these neurons, and the fact that the kisspeptin neurons sort of filled a lot of the void of where is the regulation happening. So I'm just going to give you a little bit of sort of older in vitro data that gave us some justification for making some of the in vivo models I'll tell you about. The first study just focused on estrogen signaling in GnRH neurons, and here we used two different GnRH neuronal cell lines. The GN11 cells were derived from a tumor that emerged early in embryonic life, the thought is, and that these are a immature, pre-migratory population of GnRH neurons. The GT17 cells were harvested from a tumor that was thought to develop postnatally and is thought of as more of a sort of more mature, post-developmental, post-migratory cell line. And what you can see here, this is just PCR. You can see there's ER alpha and beta in the GN11 cells. We have undetectable levels of alpha in the GT17 cells, but we do see ER beta and also progesterone receptors in both. The functional data below matches up then with what we see up top here. What we used were two different agonists, a beta-specific agonist, DPN, and an alpha-specific agonist, PPT. And you see here that both the beta agonist and the alpha agonist, alpha beta, inhibit GnRH expression in the GN11 cells, where only the DPN, the beta agonist, represses GnRH expression in the GT17 cells, not the alpha agonist, the PPT. In a separate group of studies, we were exploring kisspeptin signaling in GnRH neurons, and as part of that study, we examined the effects of estrogen on kisspeptin signaling. And the paradigm is shown here, but essentially we used the same cell lines, GT1 and GN, and we looked at both secretion of GnRH and expression of GnRH mRNA levels. And what you can see is that kisspeptin stimulates both secretion and expression in both cell lines, and that's this black bar here and this bar here, and that estrogen treatment causes a suppression of both secretion and expression, and also blunts or attenuates the response to kisspeptin. And I'm showing you this because we use kisspeptin stimulation in mice as a probe later on for GnRH function. Okay, so the aims then of this study were really to directly assess the role of ER beta and ER alpha in the GnRH neurons using conditional knockout mice. So the first model, we knocked out the ER beta gene, specifically in GnRH neurons. We call these the gerbco, I don't know, gerbco mice, I guess. And these studies were mostly performed by a Brazilian faculty member who did a sabbatical in our lab for a year, Jonas Grasselli, and he's back in Brazil now. And the second study I'll tell you about, even really in a much more preliminary state, are the ER alpha specific knockouts, or the G, this is supposed to be an GN, sorry, this is a typo. It's correct later, but the GnRH neuron-specific ER alpha knockout mice. And these studies were primarily performed by Sheng Wu, who's a faculty member in our laboratory. Okay, so I want to go back to the ER beta story first. As I said, the ER beta knockout mice exhibit a variable phenotype depending on who you're talking to. And the first knockout model from Ken Korach's lab didn't really exhibit much infertility. This was likely because these were knockouts produced sort of in the original way they produced knockouts, where they knocked in an allele basically with the neomycin resistance gene sort of still sitting there in the gene. And these mice actually were probably hypomorphic, and this was true for his ER alpha knockout mice as well. They probably had a little bit of ER alpha and ER beta activity remaining. Now, subsequent groups used a different method. They used a Krey-Lox method to really get rid of the functional domains of the receptor, and these ones, by all the methods that I'm aware of, show complete knockout, both functionally and immunogenically. And they, again, even in this model of using a knockout with the Krey-Lox system, there are differences reported in the reproductive phenotype. But it does appear that there are multiple levels of the hypothalamic pituitary gonadal axis may be impacted in the ER beta complete knockout mice in the literature. Okay, so because of that disagreement, I would say, in the ER beta knockout models, we, in making our GnRH specific ER beta knockout, wanted to make sure we were comparing it to like versus like. So we wanted to compare it to complete knockouts that we made in our own laboratory with the same basic background strain in the same colony. So here we used the Krey-Lox system with the Cree mouse with the Cree bacteriophage recombinase under the control of the E2A promoter. And this is a constitutively expressed gene, and we mated that with a floxed ER beta mouse that we produced in our laboratory, and this results then, after a couple of rounds of mating, in a complete ER beta knockout mouse. So these are the SERPCO mice, and now we're going to compare them, along with comparing them with intact, ER beta intact controls. We're going to compare our GnRH, our gerbco mice, with the SERPCO mice, okay? And these mice are made using a different Cree mouse, one that we produced that has about 3,500 base pairs of the GnRH promoter fused to the Cree recombinase gene. Same ER beta flox mice, producing a mouse in which the ER beta gene is disrupted in the GnRH neurons and intact elsewhere in the body. I feel like I missed a slide here. I did miss a slide. Where is it? It's a key one, too. Okay. There it is. Why didn't it show up? So this is really important validation of our models that I wanted to show you. So up top is genotyping we performed on the different tissues from the mice from these different models. So basically we took out all these different tissues, extracted genomic DNA, and then just genotyped them to look for the floxed allele, which is 1,544 base pairs by PCR, and the knockout allele, which is 539 base pairs by PCR. And you can see in our SERPCO that we only have the knockout allele in all the different tissues that we look at. We don't see any floxed allele, which would be a functional ER beta expressing allele in these mice. In the GERPCO mice, we only see the knockout allele in the hypothalamus. Okay, so this is, and we don't see it anywhere in any of these other tissues that we looked at, including the pituitary and the ovary and the liver, uterus. On the bottom is Western blot using an ER beta antibody that we produced in collaboration with Drew Schoenfeld at Loyola University. And as some of you are probably aware, the ER beta antibody market is a tough one. There used to be a good one, apparently, but currently there's not a lot of good options for ER beta antibodies, so this is some validation then of this antibody, and I think we have a good one here. So in our wild type versus the SERPCO mice, you can see ER beta in all these different tissues, and it's knocked out at the protein level in the SERPCO mice. In the GERPCO mice, we see retained ER beta protein really in all the tissues we looked at, including the hypothalamus, where GnRH neurons are a tiny minority of that tissue. Over here are some controls just to help with the validation of the antibody. This is in vitro transcribed and translated ER alpha, just made in a rabbit reticulocyte system, and we see no binding of this ER beta antibody to ER alpha protein. And actually there is another version of this figure where we use an ER alpha antibody and show that it does bind this protein. And ER alpha doesn't bind the beta protein, and here we show in vitro transcribed and translated ER beta binds very well to the ER beta antibody. Here's the alpha knockout. We still see protein, and the beta knockout we don't. So, okay, so now the phenotypic analysis then of these mice. And the first thing we always do is assess puberty. It's the first event of interest for us. And what we found using two different measures of puberty in mice, the first is vaginal opening. This is an estrogen-dependent process in mice. And the second one is first estrus. So this would be the first day during assessment of vaginal cytology to look at estrus cyclicity, the first time that we see estrus and the age at which we see that. And what we saw was a significant delay in both measures in the age of puberty. And there was no difference between our SERPCO and our GERPCO mice. When we looked at body weight, because puberty is very correlated with body weight in mice and most animals, actually, we see no significant differences. So these differences do not seem to be due to differences in body weight. So this suggests then that this delay in puberty is really due to the absence of the ER beta in the GnRH neuron, and so that's sort of interesting. When we looked at estrus cyclicity then in these different models over, these are representative profiles of individual mice, wild type, SERPCO and GERPCO mice. We see what looks like fairly normal estrus cyclicity in all the different lines. However, when we do why is it doing this? Okay. Hope I don't miss. When we do a more nuanced analysis of the data, we measured the percent of days at each estrus cycle stage. And what we observe is that there's a longer period in estrus, percent of days in estrus, in both the SERVCO and GERBCO mice compared to the wild type controls. And I should say the wild type controls are litter mates that don't have the Cree transgene. So they do have the floxed alleles, but they don't have the Cree transgene. Okay? So the delay in puberty and this sort of longer estrus cycle potentially is caused by a reduction in estrogen levels that we observed in these mouse models. And again, here we see it's the same between the SERVCO and the GERBCO, and it's significantly reduced versus the wild type mice. They have reduced ovarian weight, reduced uterine weight, which makes sense because they have lower estrogen levels. And these are just images then of the uteri from the different models. And this reduced estrogen levels, we think, is due to reduced gonadotropin levels in the blood. These are basal levels. Morning, we measure these in the morning. And you see reduced LH and FSH levels. And these are surge levels. They're measured an hour within the hour before lights off. So in our colony between 8 and 9 p.m. at night. And you see a significant decrease in the magnitude of the surge, LH surge, and the FSH surge, though this is not statistically significant, in our 2 knockout models. So therefore the disrupted, the late puberty, the subtly disrupted estrous cyclicity, and the reduced estrogen and gonadotrope levels appear really to be due to the absence of ER beta in the GnRH neurons. So to explore, we sort of were looking at positive feedback here. The surge is a result of estrogen positive feedback, and again, that seems to be just impaired. We wanted to look at negative feedback as well. Here we ovex the mice and we replace with estrogen, and we see that negative feedback is retained in these mice in LH and FSH. So while there does seem to be, and this is a little surprising given our basal levels of LH and FSH that we see here that are reduced. So and I'll go into that in a little more detail in a few minutes. So now ER beta is very highly expressed in the granulosa cells in the ovaries. So we would think that, my thinking going into this is that we would see a more profound phenotype in some of these measures in the SERVCO mice than we would in the GERBCO mice, and particularly ovarian morphology shown here. But really, that's not the case. We didn't. We saw reduced numbers of corpora lutea in the SERVCO and GERBCO mice, indicating fewer ovulations, and an increase in primordial and atretic follicles, sort of corresponding probably to the reduced number of follicles that were able to ovulate. This could be due to the reduced sort of basal levels of gonadotropes, but it could also be due to the lower surge levels of LH. So we're not sure yet about that. So finally, we also obviously are interested in fertility, and the way we assess fertility is we put the females with proven fertile males for 120 days and let them go, and we count we measure the day the pups are born and how many pups are born. And what you can see here is that early on, the frequency of litters are pretty much the same between the different lines, but that the SERVCO and GERBCO mice sort of peter out earlier. They have reduced numbers of litters, but you can see that it's mostly due to the fact that they're not having litters later in life, like the wild type ones. But what you'll also see is that generally the wild type mice have bigger litters than the Servco and Gerbco. So this litter, this is number of pups per litter, and doesn't take into account necessarily that they're having few litters. It's really focusing on the number of pups per litter, and you can see there's a significant reduction. And this makes sense because these mice had fewer corporalutea, they had fewer ovulations, right? So this matches up then with the data that I've showed you already. So to summarize, really the main thing I'm going to talk about today, the ER beta story, the female Servco and Gerbco mice models present with a delay of puberty, hypogonadotropic hypogonadism, which is associated with disrupted follicular development and ovulations, and a decrease in gonadotropin levels and fertility. And that we see the same phenotype basically in the Gerbco and Servco mice really suggests to us that the ER beta and the GnRH neurons plays a key role in the reproductive phenotype that we observe in the whole body knockout. Okay, so switching gears a little bit then, I'm going to tell you about some less evolved work that we have doing the GnRH ER alpha knockout. Now again, why did we do this? Partly because of our in vitro work, partly because we had the different mouse models on hand. I don't know that we expected to see anything robust in this model, and I don't know if you'd call it robust, but we do see a phenotype, so I'll tell you about that. So again, I'm using the same GnRH Cree mouse that I used in the Gerbco model, and here we're using a Phlox ER alpha mouse that we also made in the laboratory. And again, after a couple matings, you get mice that have a GnRH Cree allele and 2 Phloxed alleles, so that produces a GnRH neuron ER alpha knockout with retained expression of ER alpha elsewhere in the body. I don't know, we didn't make the complete, we don't compare directly to the complete ER alpha knockouts here. Maybe we should have, but I don't think there was much debate about the phenotype of the ER alpha knockout mice. So here, again, body weight, we didn't see any difference between the lines, and we also don't see a difference in age of puberty in the ER alpha knockout mice. The I don't know what to call these guys at this point. However, we do see that sort of same sort of subtle difference in estrus cyclicity, where we have more time spent in estrus in the knockout model when compared to littermate controls. And here's something interesting, and I'm going to spend a little bit of time now trying to explain this observation. So probably this slide and the next two. When we do the same study where we ovex the mice and then replace, we see there's negative feedbacks retained, but we see this sort of impaired ability to be released from negative feedback, or a developmental issue that doesn't give them the same capacity to secrete after release from negative feedback. But this is a significant lower levels of LH after ovex in the GnRH ER alpha knockout mice. We don't see that for FSH, which is sort of interesting. And I'm not sure quite how to explain that. That could be a pulse frequency issue, and it's something that we're actually working on in some in vitro studies and hypothalamic explant studies, but I don't have any firm data to talk about right now. Okay, so could this be because of changes in kisspeptin sensitivity then? And you know what? I think I missed another slide because I have I guess I don't. I'm sorry. I thought I had a... Okay. So could this be because of changes in sensitivity of the GnRH neuron to the effects of kisspeptin. So to probe that directly, we performed what we call a kisspeptin stim test. And it's sort of analogous to the GnRH stim test, which is pretty routine in clinical practice and in mouse clinical practice as well. So here, the GnRH stim test we also did, and this is just to show that the pituitary retains normal sensitivity to the effects of GnRH. And if you look at the kisspeptin stim test up here, we're measuring LH again. So this is sort of secondary to GnRH release, and then LH release from the pituitary. But we see there's an increased sensitivity to kisspeptin, not a decreased sensitivity. So it doesn't actually explain this data that I just showed you here, and I'm going to go into that in a second. This, however, does match up a little better with the in vitro data that I showed you before, where estrogen suppressed the effects of kisspeptin. So in the absence of estrogen receptor alpha, maybe you have less suppression of the effects of kisspeptin, or you have release from suppression of the effects of kisspeptin. So this does match up a little better with our in vitro data, but it doesn't necessarily explain our negative feedback data that I just showed you. So we can't invoke a reduction in kisspeptin sensitivity to explain this. Now, what this looks a lot like, actually, is some data that came out of John Levine's lab that I helped out a little bit with a few years ago. But they made a kisspeptin ER alpha knockout mice, and they saw a similar phenomenon in their negative feedback analysis. And they proposed that there was a defect in programming, estrogen programming of the system, and that you actually had a reduced capacity then to secrete GnRH in this line. And I don't know that that's true here in the GnRH neurons, and we're thinking about ways to test that, but at that point, that would be as good a hypothesis as any for that result. Okay, so to summarize then, the GnRH ER alpha knockout data, these mice exhibit a normal age of puberty, and so that's different from the ER beta knockout study, but they also spend a higher percentage of time in estrus, like we saw for the ER beta knockout mice. They have an incomplete response to this release from negative feedback that I was just talking about, and that's not the case in the ER beta knockout mice. And in contrast to what we might have proposed based on this, they actually have an increased sensitivity to kisspeptin. So again, the ER alpha story is a little more confusing, and we're not probably a little further away from publishing that. So really, to conclude then, the whole talk is that we think that both ER alpha and ER beta in GnRH neurons do actually play a role in regulating reproductive function, normal reproductive function. They seem to have different phenotypes, suggesting that they have different roles, which is entirely unexpected, I suppose. And we're moving forward with further analysis of the alpha mice. The beta mice, that story is pretty much ready. Actually, we're just waiting for a little histology, and then that one's done. So, okay, so I'm just going to end there. I just wanted to acknowledge a lot of folks who, over the years, were part of this study in the development of a lot of the mouse models, and folks who have recently moved to other institutions who I've worked with for a long time. Yayoi made the ER alpha mice, and I told you about Jonas and Schoen, and then our funding as well. So I'd be happy to answer any questions, and I welcome any feedback you have on this.
So Andrew, did you check out the levels of GnRH mRNA in each of them?
In the GERB, in the ER beta ones, Jonas did do that. I did not show that data. mRNA levels. It didn't match up with the levels of LH necessarily. So, in the interest of clarity, I did not show all the data that he has. But I think we will include just all of it in the paper and try to
I think
hand wave some of this stuff.
Clearly, in conditional GPR54 knockout, which I showed earlier in the talks, where the GnRH expression is way low in those animals. Like
in which mouse? Say that again.
Quarter. The conditional GPR54 knockout
Oh, yeah.
for GnRH. So then you wonder if the steroid receptors in any way participate in programming the cells to make more or less GnRH. You know, which would then
Well, that would explain
influence
the blunted
either surges or
that would explain the blunted
yeah,
response
blunted.
to Ovex too.
But it
right?
could also be timing, I wonder. Could it be, you know, in, I forget, there's some old studies of removing the time it takes for LH to peak after
Yeah.
gonadectomy.
These are a couple weeks after. So
But
we do.
the females are a little fatter, did I see?
No,
Or
they're the same
there
weight.
wasn't a weight?
Eventually, with the reduced estrogen, you might expect them to get fatter, but we did not do that one.
Okay.
Yeah. Actually, that's yeah. So that's why we also were showing the weight in the studies as well, and pretty much the same out to where we're looking. Yeah.
Okay.
And also, they do have it's not no estrogen. So I'm wondering actually if they'll have the metabolic phenotype you see in the knockout models.
So I didn't follow. So you made the knockout, both the alpha and beta knockout,
Yes.
only in GnRH neuron or globally in the entire animal.
In the beta knockout, we made a complete knockout, the
Uh-huh.
SERPCO, and we made a GnRH specific beta knockout called the GERBCO.
Okay.
For the alpha, all I showed you was GnRH, alpha knockout, and control.
Okay. So now in the beta knockout globally, apart from the reproductive functions, have you looked at the effect on the breast tissue or cardiovascular?
No, we
functions?
didn't look at it. We're a feedback, pretty much a feedback lab. Where the beta mice are available, and other groups are studying those aspects now.
Right.
And we're happy to share them also if somebody wanted them. But we that's not what we're looking at. No.
Okay. And then you mentioned that you have even a male mouse with the ER beta knockout. I think if I remember correctly, you mentioned in your talk.
I said that in the we don't look at males are boring. We don't look at them.
Okay.
But but I will tell you that in the literature, the beta knockout mice are fertile and have no reports of any reproductive anomalies that I know of. And the female, again, the female goes from slightly infertile to almost completely infertile, depending on who is looking at it. And I think even the Chambon group reported that there were sort of three classes of in their colony. There were some mice that were kind of fertile. There were some that were more infertile. They grouped them into three categories of fertility within the same colony. So
the beta ones are, well, both of them are showing longer estrus or just
yeah,
beta?
yeah, both.
So what becomes really interesting if you go back to the early debate about what ER beta was doing when Ishvan and I found the ER beta, and then Robert Steiner did a time course of how long it took for galanin to be expressed, and it's expressed on estrus. And the whole idea then, if it's expressed on estrus, was that it terminates the secretion of LH, which then would allow the cycle to reset if you're missing, and it's induced mainly by, ER beta, but maybe ER alpha has some role in that too, I don't know. It wasn't quite as impressive.
Yeah.
But this whole notion may be that the reason estrus is prolonged is you're not stopping everything to shut it down to make the whole cycle begin again. So you get stuck in estrus for a little bit longer.
Well, if you don't ovulate at the same timing, I mean, we didn't actually, I mean, the timing, we didn't look at other time points. It could be that we're just missing the timing issue.
Yeah.
And maybe
Yeah,
it's
timing
later,
screwed
and
up.
that's why estrus is longer, right? It's possible that it could be that, and it's something that we should look at.
We did look at the per one is.
Sean looked at that for the alpha one. She was doing 3 o'clock and 8 o'clock, and she was doing different times during the day,
Yeah.
but Jonas didn't do that for the beta.
Yeah,
That's something to think
I mean,
about.
that would be interesting if it was altering timing. But you've gotten nice timing.
But it appears also that beta seems to be more stimulating somehow in this model, and then alpha, maybe if it does anything, it's sort of repressing, and so that matches up in the literature a little bit with sometimes where you see co-expression of alpha and beta, that they have sort of
Yeah,
contrasting effects, but it's not always, right? So
right. And I think there is even some data out there that beta went up when estrogen got lower.
oh, okay.
Or something, whether